Hello students, welcome to EPG Pathshala. I am Dr. N.P. Singh from Center for Biotechnology, Maharshi Dayanand University, Rohtak. I am going to talk about module cell cryopreservation and animal conservation under the paper Animal Cell Biotechnology. In this module, we are going to learn history of cryopreservation, principle of cryopreservation, methods of cryopreservation, factors affecting cryopreservation, cryodamage of the cells or the tissues, applications of cryopreservation, biotechnology and conservation. Cryopreservation or cryoconservation is a process where organelles, cells, tissues, extracellular matrix, organs, or any other biological constructs susceptible to damage caused by the upregulated chemical kinetics are preserved by cooling to a very low temperatures, typically in the range of minus 80 degree Celsius, using cold carbon dioxide, or minus 196 degrees Celsius using liquid nitrogen. At low enough temperatures, any enzymatic or chemical activity which might cause damage to biological material is effectively stopped. Cryopreservation methods seeks to reach low temperatures without causing additional damage caused by the formation of ice during freezing. Traditional cryopreservation has relied on coating of materials to be frozen with a class of molecules termed cryoprotectants. New methods are consequently being investigated due to the inherent toxicity of many cryoprotectants. By default, it should be considered that cryopreservation alters or compromises the structure and function of cells unless it is proven otherwise for a particular cell population. Cryoconservation of animal genetic sources is the process in which animal genetic material is collected and stored with the intention of conservation of the breed. Natural cryopreservation. Water bears microscopic multicellular organisms can survive freezing by replacing most of their internal water with the sugar trehalose, preventing it from crystallization that otherwise damages cell membranes. Mixtures of solutes can achieve similar effects. Some solutes, including salts, have the disadvantage that they may be toxic at intense concentrations. In addition to the water bear, wood frogs can tolerate the freezing of their blood and other tissues. Urea is accumulated in tissues in preparation for overwintering, and liver glycogen is converted in large quantities to glucose in response to internal ice formation. Both urea and glucose act as cryoprotectants to limit the amount of ice that forms and to reduce osmotic shrinkage of cells. Frogs can survive many freeze or thaw events during winter if not more than about 65% of total body water freezes. Research exploring the phenomenon of freezing frogs has been performed primarily by Canadian researcher Dr. Kenneth B. Story. Freeze tolerance, in which organisms survive the winter by freezing solid and ceasing life functions, is known in a few vertebrates. These are some examples of natural freezing. So these are the examples of frozen wood frogs which has been preserved, cryopreserved in ice in a natural environment. 5 species of frogs like Rana sylvatica, Pseudacris tricerata, Hyla crucifer, Hyla versicolor, Hyla chrysoceles, one of the salamanders, Hynobis kislingi, and one of the snakes, Thamnophis sirtalis, and 3 of the turtles, Chrysimis pixta and Terrapin carolina, Terrapin ornata. Snapping turtles like Shelindra serpentina and wall lizards Podoresis muralis also survive normal freezing, but it has not been established to be adaptive for overwintering. In the case of Rana sylvatica, one cryopreservant is ordinary glucose, which increases in concentration by approximately 19 millimole per liter when frogs are cooled slowly. History. Now we are talking about history of cryopreservation. One of the most important early theoreticians of cryopreservation was James Lovelock, born in 1919, known for gear theory. He suggested that damage not damage to the red blood cells during freezing was due to osmotic stress. During the early 1950s, Lovelock has also suggested that increasing salt concentration is a cell as it dehydrates to lose water to the external ice might cause damage to the cell. Cryopreservation of tissues during recent times began with the freezing of fall sperms, which during 1957 was cryopreserved by a team of scientists in UK directed by Christopher Pauls. The process was applied to humans during the 1950s with pregnancies obtained after insemination of frozen sperm. However, the rapid immersion of the samples in liquid nitrogen did not, for certain of these samples, such as types of embryos, bone marrow, and stem cells, produce the necessary viability to make them usable after thawing. Increased understanding of the mechanism of freezing injury to cells emphasize the importance of controlled or slow cooling to obtain maximum survival on thawing of the living cells. A controlled rate cooling process allowing biological material samples to equilibrate to optimal physical parameters optimal osmotically in a cryoprotectant, a form of antifreeze, before cooling in a predetermined or controlled way proved necessary. The ability of cryoprotectants in the early cases, glycerol, to protect cells from freezing injury were discovered accidentally. Freezing injury has two aspects: direct damage from the ice crystals and secondary damage caused by the increase in concentration of solutes as progressively more ice is formed. During 1963, Peter Mazur at Oak Ridge National Laboratory in the US demonstrated that lethal intracellular freezing could be avoided if cooling was slow enough to permit sufficient water to leave the cell during progressive freezing of the extracellular fluid. The rate differs between cells of differing size and water permeability. A typical cooling rate around 1 degree Celsius per minute is appropriate for many mammalian cells after treatment with cryoprotectants such as glycerol or dimethyl sulfoxide, but the rate is not as a universal optimum. Principle of cryopreservation. Water mediates all chemical reactions in the cell. Some cells can be preserved for several months at -80 degrees centigrade. Decrease or prevent of physical and chemical reactions in the cell will take place when these were freezed at -80 degrees Celsius. All the chemical reactions are prevented under -130 degrees Celsius, which is usually preservation under liquid nitrogen, which is of the temperature of -190 degrees Celsius. Method of cryopreservation. There are two methods of cryopreservation. One is slow freezing method and the other one is rapid freezing method. In rapid freezing method, these are of further two types. These are ultra rapid freeze and vitrification. Now we will take first slow freezing. It is by means of manual freezing and by means of programmable freezing. In case of manual freezing, decreasing the temperature of the cells or semen while adding a cryoprotectant in a stepwise manner and after plunging the sample into the liquid nitrogen. The cooling rate of the specimen from room temperature to 5 degrees Celsius is 0.5 to 1 degree Celsius per minute. The sample is then frozen from 5 degrees Celsius to -80 degrees Celsius at a rate of 1 to 10 degrees Celsius per minute. The specimen is then plugged into liquid nitrogen at -196 degrees centigrade. Slow programmable freezing. Slow programmable freezing will be carried out in the following way, in which liquid nitrogen is poured into the tank and the machine, once programmed, uses the software data logging to obtain cooling from 20 degrees Celsius to -80 degrees Celsius at a rate of 1.5 degrees centigrade per minute and then at 6 degrees centigrade per minute. At completion of freezing, the straws are removed and stored into liquid nitrogen at -196 degrees centigrade. This takes about 40 minutes. The technique is simple to use and it does not require continuous operator intervention. It increases the reproducibility of the freezing operations. Rapid freezing. Using high concentration of cryoprotectants, about 40%. It is of two types, that is ultra rapid freezing, which includes ice crystal formation and vitrification. In case of vitrification, it uses cryoprotectant mixtures at which of the concentration of 40%. These cryoprotectants are of low molecular weights and high membrane permeability. The high concentration of non-permeable additives using capillaries, wire loops, micro drops, and foils, etc. The advantage of vitrification. The advantage of vitrification includes no ice crystal formation. It achieves rapid equilibrium in the cell, absence of water leak after equilibrium, decreased osmotic shock, short time required for rapid freezing process, minimum damage of membrane lipids, simple procedure, no equipment required for vitrification. What are the disadvantages of vitrification? The disadvantages of vitrification includes, for that particular process, we will have to establish a protocol. Ultra rapid thawing is required. Ice crystal formation during hesitating thawing process will be occurred. Tests for the genetic damage need to be carried out after vitrification. Cooling rate. The optimum cooling rate will be of 1 degree Celsius per minute. to compromise between fast freezing minimizing crystal formation and slow thawing for extracellular water migration. Cooling curve is affected by ambient temperature, insulation of that particular cells, specific heat of ampullae contents, volume of the ampullae, latent heat absorption during the freezing. Insulation during freezing will be carried out using polystyrene foam boxes to store the vials. This setup provides insulation for 1 degree Celsius per minute cooling. These will be placed in -80 degrees Celsius freezers, transferred to the liquid nitrogen after 24 hours or -150 degrees Celsius freezers. If at keen, just leave it in -80 degrees Celsius. Liquid nitrogen is widely used to store cells for the long-term usage in the -190 degrees Celsius freezers. Several types of liquid nitrogen cryofreezers are available for the studies. Benefits of freezing cells. There are a large number of advantages of this technique. For example, the benefits of freezing cells include genotypic drift, senescence leading to extinction of the cell lines, transformation to the tumor-related properties, contaminations, saving reagents and time, equipment failure such as incubators, and cross-contamination by other cell lines. Now we will talk about what is the theoretical background of cell freezing. Optimal cell freezing is characterized by maximum number of viable cells upon thawing, minimum intracellular crystal formation, minimum formation of foci of high solute concentration. Optimum freezing is accomplished by cooling slowly so water escapes, cooling fast enough to avoid crystal formation, use hydrophilic cryoprotectants, storing at lowest possible temperatures, minimize negative effects of solute foci on proteins, thaw rapidly, and minimize crystal growth and solute gradients. Cell concentration and freezing medium for cryopreservation. High cell concentration seems to enhance survival of the cryopreserved cells, possibly due to leakiness effect from the cryogenic damage. Concentration is avoided since dilution of cryoprotectant is high when reseedings. For example, 1 milliliter of 1 into 10 raised to power 7 cells diluted to 20 milliliter volume, giving 5 into 10 raised to power 5 cells per milliliter. If cryoprotectant was 10%, it will become 0.5%. Toxicity is unlikely at 0.5%. The residual cryoprotectant can be removed as soon as cell starts growing. The freezing medium will be used for the cryopreservation of dimethyl sulfoxide, glycerol. Dimethyl sulfoxide used at 5 to 15%, almost 10% is used as a common studies. DMSO should be stored in glass or polypropylene and can dissolve a rubber and some plastic leading to the impurities. Many laboratories increases FBS concentration to 40, 50 or 90%. Factors affecting cryopreservations. A large number of factors which affects cryopreservation includes water. Water affects in a way that it creates crystallization and volume will be increased in the cells. Physical damage to the membrane and microorganelles in the cell. It also leads to the cell death. Another important factor which affects cryopreservation is the cryoprotectants. It leads to the membrane permeability, high osmolarity, non-crystallized materials, easily supercoiling, low melting point, ethane diol, propion diol, dimethyl sulfoxide glycerol, and di- and triethylene glycols. Some other factors which includes seeding temperature, buffers, salt concentrations, non-permeable additives like sugars, raffinose, polyethylene glycol, polyvinyl pyridone, ficol, etc. And cooling rate is also an important factor which affects cryopreservation. The materials and species-specific manner, chilling sensitivity, which is also a major factor of that particular cryopreservation. Now, another important factor is what are the damages which will be done during the cryopreservation. Cryodamage. There are two types of cryodamages which will be occurred during cryopreservation. Number one is the physical and the other one is chemical damage. The physical damage will include rapid dehydration and hydration of the cells, temperature falling, shrinkage. The chemicals which are included during this particular process are cryoprotectants. How these cryoprotectants lead to the cryodamage? They raises free tubulin concentration, spindle size will be reduced, microtubule disorganization will be occurred, chromosome dispersed in 10 to 30 minutes at room temperature, microfilament disrupts, inducing chromosomal abnormality, detrimental effect on the endoplasmic reticulum, Golgi bodies, mitochondria, and cytosol. Temperature is also plays an important role to create cryodamage to the cells. Changing lipid membrane permeability, causes of the ice crystal formation, unrepairable microtubule damage, latent heat at seeding points, increased temperature, salt concentration increases, which ultimately increases outer part osmolarity and which leads to the rapid removal of water and ultimately resulted in rapid shrinkage of the cells. Salts. How salts damages during cryopreservation. Salt concentration relatively increases, which creates protein precipitation in the cells, membrane protein damage, including membrane enzymes, membrane function changes, sodium chloride, choline chloride, polyethylene glycol, etc. It also, during the cryodamage, increase in reactive oxygen species will occur, which leads to the mitochondrial damage and decreased the ATP synthesis. What are the applications of the cryopreservation? The application of the cryopreservation includes cells, tissues, and organ preservation, preservation of endangered species, genome resource banking, and, and various animal tissues and cells like oocyte, embryo, semen, ovarian tissues, tissues preservation, embryos. For maximum pregnancy achieved in a single stimulation, it decreases the risk of multiple pregnancy. It reduces the risk of OHSS, synchronization of for donation program. What are the methods which can be used for the cryopreservation of embryo? These are slow freezing, rapid freezing method, ultra rapid freezing method, and vitrification method. What are the risk of freezing of these cells? It increases the chance of chromosome aneuploidy after time, spindle fiber damage, polar body extrusion, movement of pinocytosis, cytokinosis damage, microfilament damage, zona hardening and damage, cortical granule exocytosis, relatively good fertilization and developmental rate, lower viability and pregnancy rate than the embryo, various pregnancy rate was reported, and high absorption rate. Cryopreservation methods which has been used for the oocytes are DMSO and slow freezing method, vitrification, slow freezing method, high concentration of additives and high viable rate than the conventional methods. Ovarian tissues, how these ovarian tissues can be cryopreserved. Primordial follicles in ovarian cortex, preservation of fertility to the disease animals. No ethical and legal problems has been faced during that particular cryopreservation, transplantation, and a good method for infertility treatment. Now we will talk about how these sperm and testicular tissues can be cryopreserved. The applications of these sperms and testicular tissues which are cryopreserved can be used for animals with the possibility which has the possibility to lose the fertility by certain diseases, low count of sperm, asynchronous time of ejaculation and ovulation in the process of IVF. In non-obstructive zoospermia, the testicular tissue is obtained by a biopsy and then cryopreserved these tissues. Cryoprotectants like glycerol, dimethyl sulfoxides can be used and additives which can increase the permeability of the sperm or the cell membrane. Sperm and testicular tissues cryopreservation methods were used for sperm and testicular tissues. Cryopreservations are rapid freezing by vapor of liquid nitrogen. Testicular tissues were freezed with the help of programmed slow freezing. Sperm survival rate, which affect the rapid change of osmolarity by freezing solution. Cell membrane damage by cryoprotectants. Now we will discuss how biotechnology and conservation are related. How we can conserve these germplasm with the help of biotechnological approaches. Modern biotechnology began with the first recombinant DNA experiment in 1973, but its real application starts after 1980s when the US Supreme Court held that the genetically engineered microorganisms was patentable. Then biotechnological companies formed to commercialize recombinant DNA technology. In the recent decades, a multi-centric project on bioprospecting of biological wealth using biotechnological tools have been launched in various countries. The overall objective is of characterization, inventorization, and conservation of biodiversity of different ecogeographical regions and prospecting of genes and biomolecules. This is helping in developing conservation strategies, especially for endangered species. Gene prospecting studies have also been successful, and today various stress-tolerant genes have been identified, characterized, and cloned. Studies are also going on for raising transgenic animals from these genes. Biotechnology in ex-situ conservation. Ex-situ means away from the habitat or site. Conservation method of biodiversity and wildlife protection is seldom sufficient process. Almost any species could be preserved ex situ if enough money were devoted to it, but in practice it is usually high quality species or races such as relatives of domestic animals and charismatic species which are protected in this way. In general, ex situ methods have proved a very valuable complement to in situ methods, and a number of species would not have survived without the synergy between the two. Inbreeding, depression of wildlife, and need for the germplasm conservation. Inbreeding depression is a decline in viability and fecundity of individuals resulting from mating between the close relatives. Inbreeding is minimized in an idealized populations in which individuals mate randomly. However, populations may depict from this giving a low genetic variation. If deleterious recessive alleles become homozygous, pathological conditions results. Wild populations are seldom sufficiently well known to witness inbreeding depression, resulted in high rate of mortality in their natural habitats. Conservation strategies of such population is highly essential. Thus, the use of biotechnological tools in conservation practices are very much essential today. There are some adverse impacts of biotechnology on biodiversity, especially with respect to non-target effects of genetically modified organisms in natural ecosystem. So these are the well-known examples where we can use this cryopreservation technology to raise the animals with improved traits. So, students, let us summarize the whole talk. First of all, by cryopreservation, tissue banking can be a successful key component of many different therapies and diagnostic services. More research is still needed on fundamental cryobiology, long-term outcomes after cryopreservation, on better and safer techniques. Embryo, sperm, testicular tissues are generally used and archived by frozen thawed programs. It can be used for the frozen thawed oocyte and ovum tissues. Thank you.